Okay, our next APH talk is by Dr. Karl Hasenstein from the Department of Biology, University of Louisiana at Lafayette. Karl is very unique. He's both a physicist and a biologist, and he uses both of those skills in designing his experiments. We worked with Karl originally on what's called the biotube experiment back starting in 1998. He wrote a proposal that was so highly scored in peer review that even though we didn't have hardware that could satisfy the requirements, we at Kennedy were directed to create the hardware. This is extremely unusual. He ended up using high gradient magnetic fields to cause root curvature, roots to actually curve away from the direction of the high gradient magnetic field during spaceflight on the shuttles under microgravity conditions. So very interesting as a potential countermeasure. Today, Karl will be telling you about the second peer-reviewed space biology experiment to be selected and utilizing the advanced plant habitat growing radishes. Karl. And I took it easy on you.
Which button do I push?
This one advances the slides.
Good morning. Thanks for giving me a chance to explain a few aspects of our space experiment. And I would like to point out that I'm going to be talking about two aspects. It's sort of the interface between the biology and the technology that is required to grow plants in space. And to make this work, one has to be mindful of the different constraints that are imposed on the plants as well as on the hardware. And I don't know if I should say forgive me, but I will point out a few things that are less than optimal and indicate vast space for improvement. Maybe I should say room, but I like space. So the objective of the experiment was to go in a transition from plain plant biology to a technology assessment that allows the biomass production for future crews and maybe outposts on Moon and Mars. In order to handle plant growth, we have to be able to address substrates as well as the composition of fertilizer mixes to produce in the shortest amount of time the best quality crop. And we will be talking about these aspects and to some extent to some other biological technology developments for which I will unfortunately not be able to present data, but nonetheless I point out that it is possible to assess repeatedly biomaterial using a technology called solid phase gene extraction that allows you to get a temporal assessment of transcriptome values. So, what is the timeline for the experiment that I'm going to be talking about? It started actually in 2017, and we had a launch that was fairly spectacular, as you hopefully can see here, October 2nd last year, and on a Cygnus flight or supply mission that was then captured by one of the astronauts that did our experiment for us, Mike Hopkins. And you see this on the right-hand side. Whoops. And go back. This area here that he was capturing from the cupola in the ISS. Anyways, the first reference to the complication of proper control had to do with a ground control at the Kennedy Space Center that was jeopardized. And if I tell you the reason for the difficulties, you might chuckle or cringe or something. During the ground control, a computer update was initiated that cut the power to the system for 3 days. As a result, we could not use this data set because it was really not appropriate for plants that had not seen any light for that extended amount of time to serve as a control. So this was then done as a so-called redo ground control, and because of the expenses and time constraints, the second grow out for the ISS was never replicated. So we are sort of limping along on the data related to this space/ground control experiment. Anyways, we got the material back in June and did ground control in our own lab to see how different conditions, and I will refer to this later, compare to space and KSC controls. So the plant material that we chose was radish, simply because it produces quite a bit of biomass. It is healthy, and it has all the benefits of being applicable to the Arabidopsis genomics, proteomics, metabolomics, because it's a member of the Brassicaceae. At the same time, as this picture shows, you have a tremendous variability in the formation and the coloration of these radishes. And the intent, of course, growing plants for the crew consumption was to have it tested by the crew, which turned out to be a challenge. Not for the crew, but for the administrators. So on the right-hand side, you see that radishes are extremely well, you might say, annotated, and we intend to take advantage of this. The corresponding data are being processed, analyzed as we speak. So the transition from seedlings to mature plants is a different challenge because, as we know, the requirement for nutrients, even light and light quality, changes over time. And this has to be taken into account. Nonetheless, we have only 4 weeks to grow our radishes, and that is a relatively easily managed amount of time. We have suitable substrates, and as you will see in a few minutes, the substrate is based on a compound called arsenite, and it has its own constraints, and we are working on improving this condition, replacing it by polymer beads, and you should take a look at the poster number 22 that will explain some of the aspects of changing arsenite for a more forgiving substrate for plant growth. The planting depth in these science carriers, as Howard pointed out, is somewhat of a challenge as well, because we have only 5 centimeters, 2 inches to work with, and that is still interrupted by various sensors that are included in this volume. We had developed a particular way of applying fertilizer, and we were not able to modify much of the watering system because this is an intricate part of the hardware. Nonetheless, the biological research was possible, and I will show you some of the data of this. So you have seen this before, but this is the summary of plants growing on the ISS and on the growth control at KSC. And what you should note is that there is really not much of a difference, and this is the same statement that Norman made a few minutes ago. The plants behave quite similarly, which is good, but we have to look a little closer to see if that is indeed holding true when we come to analyze the metabolites that are of interest for nutritional assessments. You may also have seen that we collected leaf punch data from the plants as they grow in order to assess the temporal element of plant development. And I will be able to tell you a few things about that. So, back to the statement that we have a state-of-the-art piece of equipment available at the ISS APH, and I would like to point out a few of these things. The first cultivation condition that I'm drawing your attention to has to do with CO2. The ISS atmosphere is about 5,000 parts per million CO2. We decided to tone it down a bit, and the intent was to grow the plants at 2,000 parts per million and compare this to what we have on the ground, which is roughly 400 parts per million. So while the attempt was made to have a constant CO2 content, these data clearly indicate that this is not the case. And you have some variability in between the two grow-outs on the ISS. It is unclear why this large fluctuation occurred, but it is also observable on a daily level, which is what you see on the bottom of these slides. So this is not conducive to have a good assessment of these effects. The second element concerns light. This is critical for control. So when you look at the growth control at KSC, stay on this one, you see that there was a snafu maybe, and the light conditions for the first maybe 4/5 of the experiment were significantly lower than what we had on ISS. At the same time, we also see significant variability in the data during the grow out, and they were not the same, temporally speaking, between ISS1 and ISS2 grow outs. This is important because we have to consider the fact that things are just not as predictable as one wants them to be. The most complex system has to do with the water supply. The data that you look at here show you the traces of moisture sensors within the root zone of these science carriers. And what is important is that on the initial period of water supply on the ground, that is to say the KSC ground control, you have a clear distinction between the upper and lower sensors. The lower sensors are the ones that you see in the tracings on top, have a higher concentration of water, you might say. This goes away in space because these traces are essentially the same, which is good to know. However, the supply of water is different in the two configurations, and that is probably one of the reasons why the plants on the ISS did reasonably well. So the fluctuation that you see later is the attempt of the system, the computer, the sensors, the dispensers, to catch up with the water demand of plants. And as they grow, transpiration becomes really significant, and a large number of water needs to be funneled, so to speak, through the plants and recycled. And this is a strain on the system that is shown by the variability of these traces. When we look at the outcome of the experiment, I show you here 4 different values of biomass for the bulb, the actual radish, and the leaf area, because the leaf area is related to the ability of a plant to produce metabolites. And what you see is that the two, first off, I should say that there is no statistical difference between any of the grow outs. But the indication seems to suggest that the ground controls fared a little bit better than what we had on space. Nonetheless, the overall outcome was quite similar to what Norman had explained earlier. We looked at the leaf area and we saw the same trend. What is a bit uncertain is the following thought experiment. If you take the leaf area and you divide it by the bulb mass, then you should get an assessment of the quality of the performance of the leaves. And in that case, the ISS experiments proved to be superior. But it is still uncertain whether this is the result of the elevated CO2 concentration or not. So, when we look at the crude survey of metabolites, we can say that the nitrogen and mineral distribution between the grow-outs on station and at KSC and even in our own lab are quite comparable. The point is that the leaves have higher nitrogen content than the bulbs. The leaves have higher mineral content than the bulbs. And for that reason, I encourage you to not throw out the leaves of the radishes, but eat them as a salad. They're more nutritious than the radishes themselves. So, going on, we look a little bit at the aspect of biology and the photosynthetic capabilities. And we used the capability of the hardware, of the APH hardware, to assess the reflectance in the red and the far red light of the plants. Unfortunately, the camera is positioned laterally so that we cannot get a complete overview from the top. Nonetheless, as the plants grew, you see that the reflectivity in the far red was significantly higher, and this allows us to get an assessment of the so-called normalized differential vegetative index. And the diagram on the right indicates that we have a trend that unfortunately goes downward, meaning that the photosynthetic efficiency is not as high as it could be or was at the beginning. And interestingly, the data from KSC seem to be a little worse than the ones on ISS. That goes in parallel with the statement I made earlier that the overall effectiveness of the leaf material is higher at the ISS than in ground controls. So moving on to the temporal analysis of the leaf punch data, we were able to collect this at day 10, 18, and 24 out of the entire grow-out. The intent was to see how different genes that have particular metabolic relevance for radishes fare over time. And you see the outcome here that indicates that in some cases you get a steady decline, and in other cases a fairly constant expression of this particular transcriptome. I need to point out that these data are in reference to two reference genes, actin and ubiquitin. This is the 0 line. But interestingly, this is not shown here. The reference genes are not that great as a reference gene because they show some fluctuation as well. But since they are expressed at to a far greater extent than the individual genes that we looked at here, it's still a valid assessment. So we have some information, and this is not the whole story, of material changing, of the transcriptome changing over time. Work in progress. We are looking at the sugar content of the plants, mostly the bulbs, because they have an element of the flavor. The second thing that is relevant is the glucosinolate analysis, and you see some generic assessment on this table here, which again indicates that we have a similar pattern for the ISS and the ground controls, and it confirms that the glucosinolate content of leaves is somewhat lower than of the bulbs. And the reason for that is that the bulbs, the radishes that we typically eat, are a bit more pungent. This was maintained in space conditions, and that's reassuring. We also intend to do this BG analysis that I introduced at the very beginning. Unfortunately, I'm not able to show you data. In progress is also an RNA-seq assessment of the entire comparison between the two ground controls and the ISS experiments. And the reason for these RNA-seq experiments is that we can compare the effect of elevated CO2 to normal CO2, and of course the effect of microgravity to normal gravitational conditions. But we can also look at the different outcome between the growth control without elevated CO2 in the two different locations, or I should say 3 different locations. Lastly, and this is sort of a take-home message from all the data that I showed you, and that is that the fidelity of space experiments is inversely correlated to the duration of the experiment. In other words, as longer as it takes to grow something, the more the variability may matter. So before I stop, I wanted to introduce 3 additional brief points. And that has to do with the plasticity of the biological material. What you see on these pictures are radishes that were treated in a very simple way. They were simply frozen. And when you freeze a radish, you can do this at home and you might be surprised. And you thaw this radish so that you can process the tissue. You will note that the anthocyanin of the epidermal layer diffuses into the radish itself. This is this portion of the picture. If you shave the radish before you do this experiment, of course it doesn't turn red. There are 2 aspects to this. It's not newly synthesized, the anthocyanin. The anthocyanin is not evenly distributed because you have this pattern formation right here. And lastly, the diffusion occurs very quickly. If you shave these radishes only halfway, you see that the portion that is stained is the one that was not shaved. Why do I point this out? Because the processing of data is susceptible to the mixing up of messengers, and messengers includes metabolites as well as RNAs, mRNA, short RNA, non-coding RNA, you name it. All these things tend to be mixed up, and this is important for an analysis of metabolomic pathways. The second point that I would like to make is symbolized by these pictures. What you see is a high concentration of starch grains in the top portion and the complete absence of starch grains on the bottom. What we are looking at here is a crude cross-section through the bulb tissue of radishes. And this can be investigated further. As Norman pointed out, every good research opens another question. The questions in this context are, is the elevated CO2 translated into a higher starch content? This was not on our horizon when we started this experiment. Or secondly, is this expansion, this potential expansion, limited to just a subset of these radishes? And we don't know the answer to that. But it's interesting to investigate this further. Lastly, when you take a look at the elevated CO2, it becomes critical to examine whether the differentiation between 13C and 12C, the 2 isotopes of carbon that are used in a variety of contexts is affected. A collaborator, a friend of mine, showed some time ago that as you increase the CO2 concentration, you decrease the ability of plants to differentiate between the two isotopes of carbon. So we put this to the test, and we learned a surprising lesson. The lesson is shown in this table, and it simply shows you this delta C13 information. And what you see is that we have the regular stuff, I would say what we associate with our grow out in our lab, which is dependent only on regular air. And that is the expected range of negative roughly 28 to 32 parts per million of the delta C13. When we go to the experiments on the ISS, we see that the number jumps up to negative 42 to 45 parts per million. And when we look at the KSC experiment, it is reduced to only 15. The reason for this is that during the compression of CO2, mechanisms are in place that affect the concentration of the two isotopes. This was unknown before, but it gives a valid and important piece of assessment of the quality of CO2 that is used in the APH. And I think this leaves further questions to be studied. I would like to end my talk by giving the big thank you to all the people that are listed here. And the ones on the picture is Howard, the two astronauts that took care of the experiments on the ISS, and Dave Reed from TechShot, that was an extremely valuable go-between myself, our lab, and the ISS folks. I should say also the NASA folks. And lastly, I point out my postdoc and co-author, Susan John. She's looking for a job, and I can assure you, you will not find a better person than her to do any kind of research related to plant biology in space. Thank you very much.
Okay, I'm back on. Thank you for that wonderful talk. We have time for one question. We actually got a comment from Venkat saying that it seems microbiological influence on the growth of plants needs to be explored. In other words, microbes of rhizospheres or microbial byproducts should be explored for good plant production. Would you be able to comment on that?
Yes, the growth in space is confined and depends on ventilation that is quite specific to the hardware. As a result, you have most likely a different expression of microorganisms at the beginning when the surface is smooth and the end. However, we tested this in the growth in the SVT and EVT, and it was acceptable. Acceptable is a stretchable word because it is almost impossible to meet NASA standards, but casually speaking, we outperformed what you would find in your favorite supermarket. So in this sense, it is quite good, but it is not in all cases up to snuff in terms of NASA requirements. That's as vague as I can be.
That's perfect. Thank you. Let's go ahead and thank our speaker again for a wonderful talk.